Good day. In this presentation I will be discussing gene therapy, its various forms, issues, potential benefits, and detriments to global health. So what is gene therapy? Gene therapy is an experimental form of treatment that uses gene transfer of genetic material into the cell of a patient to cure the disease. The idea is to modify the genetic information of the cell of the patient that is responsible for a disease and then return that cell to normal conditions. It is a transfer of genetic material. It is done commonly by using viral vectors that use their own biological capacities to enter the cell and deposit the genetic material. Both inherited genetic diseases and acquired disorders can be treated with gene therapy. Then there are two types of gene therapy treatment. First is somatic cell gene therapy. This involves obtaining blood cells from a person with a genetic disease and then introducing a normal gene into the defective cell. This type of gene therapy does not prevent the disease from occurring in the next generation because it does not affect the sperm and egg cells. Somatic cell gene therapy only affects the other body cells. Somatic cell gene therapy has to be done several times over the course of the patient's life because the effects do not last very long. Then the second is the germline therapy. Takes place in the reproductive cells. It involves the genetic modification of germ cells that will pass the change on to the next generation. This type of gene therapy only has to be done one time to be permanent. One type of germline therapy is to treat a pre-embryo that carries a serious genetic defect before it is placed back in the mother by in vitro fertilization. Another germline therapy is to treat adult sperm and egg cells so the genetic defect is not passed on to children. Gene therapy has potential benefits that can cure, stop, or slow disease. First, it is a treat for cancer. A variety of efforts are underway to use gene therapy to treat cancer. Some types of gene therapy aim to boost the body's immune cells to attack cancer cells, while others are designed to attack the cancer cells directly. One way the body protects itself from cancer is through T cells, a main component of the immune system. But some cancers are good at avoiding these protection mechanisms. A new form of immunotherapy that uses specially altered T cells to more specifically target cancer is called a chimeric antigen receptor, or CAR T cell therapy. Second is for vision problems. As the disease progresses, patients experience gradual loss of peripheral and central vision, which can eventually lead to blindness. Gene therapy for some IRI patients, delivering a normal copy of the RP65 gene directly to the retinal cells at the back of the eye, using a naturally occurring virus as a delivery vehicle. Third is for movement problems. For children with the genetic disorder spinal muscular atrophy, where muscular dystrophy, motor nerve cells in the spinal cord are damaged, causing patients to lose muscle strength and the ability to walk, eat, or even breathe. It replaced the missing or non-working gene with a new, working copy of a human SMN gene, helping motor neuron cells work properly. Then fourth is for infectious diseases. Researchers believe targeted gene therapy and gene editing may have widespread application for a range of infectious diseases that aren't amenable to standard clinical management, including HIV. Although HIV isn't a hereditary disease, the virus does live and replicate in DNA. Gene editing is an approach that precisely and efficiently modifies the DNA within a cell. In this approach, gene editing can knock out a receptor called CCR5 on immune cells used by HIV to enter and invade cells. Without CCR5, HIV may no longer invade and cause disease. Lastly, used for rare diseases. Fatal diseases uses gene-modified blood stem cells with a goal of permanently correcting the underlying cause of disease. Blood stem cells are taken from the patient and corrected outside the body by introducing a working copy of the gene into the cells. The gene-corrected cells are then put back into the patient to potentially cure the disease. Gene therapy has some potential risks like an unwanted immune system reaction. Your body's immune system may see the newly introduced viruses as intruders and attack them. This may cause inflammation and, in severe cases, organ failure. If it targets the wrong cells, viruses can affect more than one type of cells. It's possible that the altered viruses may infect additional cells, not just the targeted cells containing mutated genes. If this happens, healthy cells may be damaged, causing other illness or diseases, such as cancer. Infection caused by the virus. It's possible that once introduced into the body, the viruses may recover their original ability to cause disease. Then there's a possibility of causing a tumor. If the new genes get inserted in the wrong spot in your DNA, there is a chance that the insertion might lead to tumor formation. Because gene therapy involves making changes to the body's set of basic instructions, it raises many unique ethical concerns. The ethical questions surrounding gene therapy include: 1. How can good and bad uses of gene therapy be distinguished? 2nd, who decides which traits are normal and which constitute a disability or disorder? 3rd, will the high costs of gene therapy make it available only to the wealthy? 4th, could the widespread use of gene therapy make society less accepting of people who are different? And 5th, should people be allowed to use gene therapy to enhance basic human traits such as height, intelligence, or athletic ability? Thank you for watching. This presentation is made by Team 3 of BSHM Section C, including the following names: